Hi everybody. Let's continue on with this video to complete glycolysis. So we're going to talk about phase 3 that we have not discussed so far. So here we're going to have phosphate transfer, and in the first reaction from the 3 to the 2 position of glycerate, we finished off phase 2, with a 3-phosphoglycerate formation and generation of 1 ATP. And we'll see that this phosphate transfer is important to set up the formation of phosphoenol pyruvate. We're going to see a dehydration at that point. And that is going to be our high energy compound, phosphoenol pyruvate, that will be enough to drive formation of ATP from ADP. So these are the 3 reactions. First, we're going to move the phosphate from position 3 to position 2. Then we're going to dehydrate this compound, glycerate 2-phosphate, to make PEP, phosphoenol pyruvate. And finally, pyruvate kinase will come in and give us 1 ATP. Again, all these are 3 carbon units. Okay? So the first reaction is catalyzed by phosphoglycerate mutase, PGM. And as I said, all it's going to do is going to move this phosphate from this position to its neighbor carbon. Okay. However, in reality, it doesn't actually grab this and move it over here. It does something very interesting. Now, as a group, where does that fit is, of course, an isomerization. Okay. Mutases in particular, they do shift functional groups from one position to another within the same molecule. But as you'll see, they do it in an interesting way. First, the enzyme itself is phosphorylated. Okay, then it swaps phosphates. There is an intermediate that's phosphorylated at 2 positions, and the phosphate that was on the enzyme will stay on the substrate, and the enzyme itself, it regenerates itself by picking the pre-existing phosphate on the substrate. Right? So this is what I'm talking about. This is our reactant, glycerate 3-phosphate, and I have color-coded the 2 phosphates. Hopefully, it's going to make sense here. And so we can tell them apart. The one on the on 3-phosphoglycerate is shown in red. The one on the enzyme, okay, is shown in blue. So the enzyme is phosphorylated. Now, what's going to happen is that there's going to be a general base catalyst here, and this position, the 2-hydroxycarbon here, is going to pick up the blue phosphate from the enzyme. So you form this intermediate, okay, that's actually phosphorylated at 2 positions, both the 2 and the 3. Okay? And the enzyme now is not phosphorylated, and as you can guess, it will pick up the red one and become phosphorylated. So there's an actual swap of phosphates on the enzyme, and you end up with the substrate now becoming a product that's phosphorylated at position 2, okay, shown here in blue. It's actually kind of cool to decipher this mechanism, because you can see how you can label preferentially one or the other phosphate and follow where they end up. Okay. So now we got glycerate 2-phosphate. And why is that? Because it will enable phosphate activation in the next step. So it's setting up the next reaction. Okay, it's a necessary thing. Redox-wise, we haven't changed anything. In fact, all we did is and we moved this phosphate 1 position over. This is not now a high-energy phosphate. We have to make it a high-energy phosphate, as you will see in a minute. But redox-wise, all the carbons are the same as they were before. So now the next enzyme comes in and will make this phosphate at position 2 a high-energy phosphate. The enzyme is called Enolase, and what it will do is it will remove this proton and this hydroxy group, basically water, and make the double bond here. And that, as you will see, will make this a high-energy phosphate. So that's an elimination reaction. What do we eliminate? We eliminate water. Right? The mechanism is shown here. Okay, this is the 2 position. Okay, so that's the carboxyl group that we have on 2-phosphoglycerate, and we want to remove this proton and this hydroxyl group. Okay, so the enzyme will line it up, In this way, first it's going to pick up a proton, so it's going to happen in two steps. First, the proton gets removed, and then in the end, the hydroxyl group is going to be removed. So you remove the proton, and the carboxyl group here serves as an electron sink in this case. Okay, so to facilitate, I mean, these electrons, once you pick up the proton, they have to go somewhere. So you have to find a place where they can bounce on, and that's really this group here, the carboxyl group. So, and then they bounce back into the molecule. Okay, so you move this double bond here to the next position over. Okay, so you generate a double bond here, so they come in. You can follow the electrons from this oxygen. They regenerate the carboxyl group, and the double bond goes next to it, moves over to the next position. And for that to happen, the hydroxyl group has to leave also. Okay, and it does. And you remove. That's how you remove water, and you have built phosphoenol pyruvate. So again, anytime you involve these electron movements, the electrons have to go somewhere. So you have to give some groups, some features of the chemistry that facilitate, you know, the bouncing of the electrons. In this case, it's the carboxyl group that does it. It's absolutely essential to activate its dehydration. That's an important enzyme. You inhibit it every day when you brush your teeth with a fluoride-containing toothpaste because it competes with 2-phosphoglycerate. I mean, of course, the bacteria on our gums, they do a lot of glycolysis, and to survive, that's an important thing. And fluoride, it turns out, competes with glycerate 2-phosphate, so the bacteria cannot do glycolysis and hopefully die. They cannot proliferate. All right, so why again? Why? Because we really want to activate this phosphate bond here so it can move eventually onto ADP and make ATP. And the only way to do it is to really have, or one way to do it is to have an adjacent double bond to it. So why is that? Because we have this double bond. If it's not phosphorylated, for example, in this case, you will, the equilibrium will really prefer to be like this. That's pyruvate, by the way, okay, as opposed to this. So it's much more favorable to go towards the left in the equilibrium. And that's what's going to happen. However, if you stick a phosphate there, and that was the purpose of phosphoglycerate mutase, to stick a phosphate at this 2 position, it cannot go back. So it's stuck, very itching, you know, to leave. It wants to leave. It's an unstable, high energy compound, phosphoenol pyruvate, and it really wants to live. And this is what we achieved here. We generated a high energy compound that this phosphate wants to live. It's perfect. A phosphometabolite, you know, with enough high energy that wants to give this high energy phosphate to something, let's say ADP, and make ATP. In addition, phosphoenol pyruvate, as I said, it's a very high energy compound, and it's a precious building block. It's used, as we will see, much in the way we saw with 3-phosphoglycerate to make serine. So this is used for other amino acids, and especially aromatic amino acids. Okay, you really get actually not just one but 2 PEPs are used to make one aromatic amino acid. So it's a very useful and expensive building block. So, which again fits with the whole idea of glycolysis. What is it there for? To get you some ATP and also to have these key intermediates. The redox-wise, I had to imagine here a little bit. So it's intramolecularly that the redox state has changed. This carbon, you know, which will be this carbon here at the very end, okay, became a little more oxidized. Sorry, a little more reduced compared to the hydroxycarbon. Okay, it's not fully reduced like a hydrocarbon, it's not CH3, so it's CH2, so it's somewhere in between the hydroxycarbon and the hydrocarbon. So I picked gray, and the carbon next to it is a little more oxidized than a hydroxycarbon, right? So it's intramolecular, no net change, but you can see a relative change in those carbons, which will be pushed further in with the next reaction. So here, which is pyruvate kinase, and again, this is a high energy phosphate now, or phosphoenol pyruvate. It will be picked up by ADP to make ATP, and of course you end up with pyruvate. So it's the same mechanism as we have seen before. It's a group transfer, phosphoryl transfer, same as before the mechanism, how it happens, and why. like the previous enzymes that we have seen in the lower part of glycolysis, you make ATP, of course, but also that too, as it turns out, is a building block. Pyruvate is a building block for a variety of amino acids. Alanine, leucine, isoleucine, lysine, and valine, as we will see at the end of the class when we talk about biosynthesis. Fitting, again, with the general theme of what is glycolysis there for. It is also a highly regulated enzyme, and often with the same effectors that we have seen with PFK, the other enzyme in the upper part of glycolysis. For example, if there is a lot of AMP, which again, what does that mean? It means that there is a low energy in the cell, so you better start making ATP. That's a positive effector, okay? Allosteric effector, which makes sense, okay? This enzyme makes ATP, so if you don't have enough ATP, and that's the sentinel metabolite is AMP, it binds to it allosterically and activates a pyruvate kinase. On the other hand, if you have a lot of ATP or other byproducts of pyruvate, which as we'll see is it's going to be acetyl-CoA, for example, or alanine, which is pyruvate you use a lot of pyruvate to make alanine, meaning you have a ton. Those will tend to inhibit pyruvate kinase. It is very important for tumor growth. Pyruvate kinase was one of the first metabolic mutations. So mutations in metabolic enzymes, we'll see a few others that with the completion of the genome projects and so on and so forth, they found mutations in metabolic enzymes that can actually drive, not just necessary, but they can drive tumor formation. So this was published a little over 10 years ago. Pyruvate kinase, as I said, is a highly regulated enzyme. I showed you some effector metabolites. More recently, serine is also shown to be an effector, so a positive effector, so we have to add serine here too. Okay, so this is how the field evolves. I feel like I have to tell you, however, that, so it has other roles too, sorry, I will continue the next slide for the role in tumor formation. It has moonlighting roles. Okay, again, it can act as a transcription factor, but in this case, actually uses the catalytic activity. So it's not independent of its catalytic activity. It will go on and phosphorylate basically another substrate. Okay, it will go on and phosphorylate histone, and that is thought to activate transcription and maybe tumor formation. So this is a little different than the previous moonlighting roles. It's basically it uses a different substrate in this case, but it's still, for this activity, it relies on its catalytic activity. It's a hotly contested area, whether really mutations in a pyruvate kinase predisposed to tumor formation, tumor growth. I showed you the previous paper that said, yes, it does. Some others have questioned it. Okay, so the jury is still out, I guess. We have to wait for a few more studies and we'll know for sure. So that completes now all the glycolysis steps, okay? So we recovered our investment here. We got 2 ATPs from one molecule of glyceraldehyde 3-phosphate. Of course, one glucose will give rise to 2 of those molecules. So we got 4 ATPs per glucose. But of course, we spent 2 to get to this point. These are the 2 ATPs we spent to put the phosphates at each end of glucose. So 4 minus 2, we have a 2 ATP net production from one molecule of glucose. All right? And we end up with this oxidized byproduct. Okay, so we broke down. This is really, it epitomizes catabolism. Sorry, we break down, we make key intermediates that we need for biosynthesis. Let's say we got some energy in the form of ATP. And of course, in the process, we got smaller things from what we ate. Our food got broken down. That's a catabolic reaction, and more oxidized. So we got some electrons out of it. Right? And the redox state of pyruvate is shown here. We have a carboxyl, we have a carbonyl carbon, and we don't have a ketocarbon, and we don't have any hydroxycarbons anymore. We just have a hydrocarbon at this stage. So that fits with the scheme that I was showing you earlier. All right, so we will close this video with that completes phase 3 of glycolysis, and in the next one, we'll see how fermentation happens.